over the years, we have enjoyed bringing experts in the field in from different areas of health to come in and teach us about things that might be able to improve our health. And as you all know, type 2 diabetes and Alzheimer's disease, obesity are just escalating throughout not only our country but other countries. And so I'm really pleased to present Dr. Don Graves today. Don is one of our Life Fitness Center students, in addition to being an expert on cinnamon. He's also a marathon runner and just completed a half marathon in November, if I'm correct.
Right.
The Santa Barbara Marathon, and ran a half marathon. Is Margie here today?
No, she's not. She's in a bus coming back from the Huntington Gardens.
Oh, sounds nice.
So, yeah.
But they ran the half marathon in November together. So, and do you mind if I tell them how young you are?
That's fine. Yeah, go ahead.
At 74 years of age. Sorry. So, pretty amazing. I think he loaded up on cinnamon.
That's right. Well,
and then went after it. So anyway, Don was kind enough to donate his time to come and talk to us today about a lot of the research that he's doing. Don was is a professor emeritus from Iowa State, where he taught for 39 years in molecular biology. Is that right?
Biochemistry, basically.
Biochemistry. So he's a biochemist. He understands all the little nitty-gritties of how those cells work and what helps them to function. And then has been here in Santa Barbara for
7.5 years now.
7.5 years. So you may know also he was recently featured in the Santa Barbara News Press doing quite a bit of research on Alzheimer's disease, and part of the running of the half marathon was to raise some funds for some of the research he's doing with Alzheimer's disease. So a very, very active person in our community, and somebody who has, I think, made really major contributions to our understanding of type 2 diabetes and how we might be able to really try to combat what looks to be a very powerful epidemic for us. So without any further ado, I will go ahead and introduce to you Dr. Don Graves from UCSB, who's an adjunct professor there.
Wow. Thank you.
I forgot the most important part. He prefers to be called the Cinnamon Man of Santa Barbara.
That's right. That's right. Exactly. And you can see I'm dressed accordingly.
I know.
See? And the best part of the talk is this title, and that was done by Ellen. And we'll try to tell you a little bit about this connection, supposedly there is one, on cinnamon, diabetes, and Alzheimer's disease. And some of the work we've done, but a lot of work has been done, of course, by others. And we're still, even though I'm retired, still trying to do something at UCSB, but it's a good way to spend my time. So we'll just get into first a little background about cinnamon, and then we'll talk a little bit about insulin. And kind of the point I've been thinking about this a lot is we have not enough insulin, that's a problem. If you have too much insulin, it's a problem. If you have not enough glucose, that's not good. Too much glucose, that's no good. It's kind of almost like this. If a little bit of something seems okay, but too much, like you put in too much weight. I notice that when you do the exercise. See, that's why I do a little bit less weight. See, too much, you gotta be careful. I have to watch. That wasn't planned, it just sort of happened. Well, anyway.
Don follows Dale in the super circuit at 6 a.m.
That's right.
That's the deal.
So we'll go to start a little bit about a background of cinnamon. And it's been known for a long time in Chinese medicine. Thousands of years ago, they've used cinnamon. In fact, they're probably still using it. But I gave a talk about this in China, and they were really excited when I said cinnamon, but they didn't know what I was talking about. But we went out and looked at the dictionary, and they said, oh, yeah, they knew what it was. And yes, that was a big start. And it apparently, had some success. Well, we don't really know that, actually. But it was used to treat many things, and one of which was diabetes. In ancient Egypt, it was used medicinally, a flavor for beverages and in perfumes. And it seems Cleopatra used this in a way to entice Mark Antony, when he came to see her. And then for embalming. And then there's Nero, the Roman emperor, He used a whole year's supply of cinnamon, burned it on his wife's funeral pyre. He seemed to be remorseful, but he actually murdered his wife and his mother before that. So I don't know what was going on quite there, but that's what, he did use all this. Now, cinnamon has been used in all sorts of savory dishes, started in Europe, I guess, and probably other places too. In ancient world, cinnamon was considered more precious than gold, but I don't know, today with the price of gold, I don't know if you really say that. Nevertheless, you know, sometimes you would say, you know, all the money in the world might not save you if you have a real serious health issue. I'm not sure cinnamon can really do that to save you, but, you know, sometimes money isn't the only thing that counts. And on the side of this slide is a picture of a cinnamon tree. It's a pretty big tree. It can be 30 or 40 feet in height. Depends on the types of cinnamon that we're going to be talking about. But I'll show you a picture actually of a cinnamon tree in Santa Barbara. It's a little guy, and I have one like that in my garden too, and it's actually not even doing as well as this one. But this is actually this gentleman here is Norman Beard. This is his nursery. This is a tree that is the authentic cinnamon source that's found in Sri Lanka, earlier Ceylon. Oh, there we go. Okay, so cinnamon or cassia belongs to a family called Lauraceae. True cinnamon, I'm not going to go back. Thanks. Too many buttons here for me to handle. True cinnamon, Ceylon cinnamon, is known as Cinnamomum xylenicum or Cinnamomum verum. If you buy cinnamon in Europe, it's always going to be this. If you buy cinnamon in America, it's unlikely to be this. This is very expensive cinnamon, except we do have some variety of it that's apparently has come from Mexico. It's not the high quality Ceylon cinnamon, it seems. But there are other that are called cinnamon, but they're actually cassia. And Chinese cassia is one thing. And I'll show you, here's some sticks. This is not something you want to chew on, though those other sticks are much smaller. And usually you don't find Chinese cinnamon sold as sticks, but as powder. Then there's Indian cassia, Indonesian cassia, Vietnamese cassia. Now they're all very different in their compounds, and they have different properties that are important for all sorts of things. So here we see the Ceylon cinnamon in the stick form and being ground up, and there's Indonesian cassia. And here is obviously somebody that's actually working on a stick. You peel off the outer bark because that's not where the cinnamon is found. It's in the inner bark. And really, I've never really seen exactly how they do this, but it would be interesting sometime to make a visit to see how they actually prepare these cinnamon sticks. But this is obviously something that's a pretty big stick and is going to probably be ground up. So now we're going to talk about some medical claims for cinnamon. These are some things that we actually know about cinnamon and what it can do. It's certainly a powerful antioxidant. It's also can be used to relieve hypertension. It's also a strong metal chelator, and though I didn't put that on here, that's actually quite important in binding sort of toxic metals, so that's another important role. Anti-cancer activity, anti-inflammatory property, antimicrobial. Interesting about boosting of brain function. We'll come to that. And last, insulin-like activity. This is how I got to be involved with this. I got involved with this actually because I had a lot of good students in my laboratories for 39 years doing their advanced degrees, and one of them name is Richard Anderson. He's now at the USDA, and he found actually that there was something in cinnamon. He was looking for biological chromium, but he found out that cinnamon didn't have the chromium, but it had something else that had the insulin-like activity. Well, we joined up together, and we did a lot of work at Iowa State University trying to find out how the cinnamon works. but Richard Anderson at the USDA was the one that discovered that cinnamon has insulin-like activity. And this is also from his laboratory. Insulin-like activity is found in water extracts of cinnamon, but other spices as well. In concentrated extracts, many spices showed insulin-like activity, but cinnamon was the most active of all. So what are these compounds? They're water-soluble, and I'm going to show you a structure. I know you probably will cringe a bit here, but don't worry, I won't be doing too much of this. Oh, I guess it's following after this. But anyway, before that, they're known as proanthocyanins, or commonly as tannins. I think people know about tannins, condensed tannins. They're found prevalent in red wine. It's the material that gives you this astringency, this dryness that you experience on drinking red wine. Again, as I said, they're strong antioxidants, but they also have properties that bind to proteins very tightly. And there's proteins in your saliva that are sort of untangled, unfolded proteins, as we call them, rich in a certain amino acid called proline, and these compounds bind very tightly to that. And you'll see, actually, because of this, led me to start some work on a protein that's involved with Alzheimer's disease, which also had the same kind of characteristic, unfolded and rich in this amino acid proline. Okay, so now, just to show you, this is some work that we did here in conjunction with, it was supported by the Cottage Hospital. Richard Anderson was the key person, however, at the USDA, but we did some work here to lead to this structure. It's actually made up of the units where there's 3 of these connected, another 3 of these, and another 3 of these. So you make polymers. And it's got a lot of these OH groups on it. These are where the metals can chelate. These are where the antioxidant activity can take place, and electrons can be transferred all around here and trap that from doing the damage in cells. But then they can bind to proteins through all of these structures. So they're really quite interesting molecules. And we've been testing lots of these, and some small, some large, to try to find out what's the best one for our biological experiments. So now I'm going to talk a little bit about diabetes, insulin, and what's called protein phosphorylation. Okay, so today there's a large 20.8 million people in the United States have diabetes. The major fraction of people that have diabetes is not what's called the type 1 diabetes, which is where the beta cells of the pancreas are destroyed and that you need insulin. The most prominent one is where we have a non-insulin dependent, or what's called type 2 diabetes. It used to be called adult onset diabetes, but that's really not true anymore because even children are showing now this type of diabetes. It starts out actually where the insulin actually becomes to a higher level than normal, and that the cells that kind of respond to that higher level of insulin and become resistant to the insulin. And though even you have the insulin there, it isn't really working well. So it's quite a different case where in the type 1 you have to have the insulin. In the other case, you got too much of it, and something happens and turns off the cell machinery. Now, there are certain things we can do to help ourselves along, to help from getting type 2 diabetes. And you talked already about importance of exercise, diet, and we really know quite a lot about exercise, for example, and diet, and how this can influence it and overcome, by exercise, some of the problems of insulin resistance itself. So it's really important to be physically fit all your life. Start now, you young people here, and for us too, it's never too late. So there are lots of serious health consequences. We're not going to go through that, but you can imagine one of the problems really comes from glucose. Now, glucose, which we need so much for our energy, and it's important. But if we get too much glucose, then we start seeing effects all throughout our body, destroying the tissues, binding the proteins, and cross-linking them so that the tissues don't function correctly. You see that, people with diabetes and vision problems. But it's all over. It's all over. And you see the same effects of hyperglycemia in the brain. Now, I really want to get to this insulin part more in the brain, and we'll go through that. So I guess we'll just hold off a little bit because we've got some slides ahead on that. But we do know that there is a connection between insulin resistance now and type 2 diabetes, and this all may be connected some way with Alzheimer's disease. I'll try to give you some of the work that's been published by other people on this and why we got so excited about it because of our work on diabetes with cinnamon. So insulin is a protein that comes from the beta cells of the pancreas. Oh, I should say that, okay, we're not showing structures here, but to understand, insulin is a protein. The material in cinnamon is not a protein. It has the properties to act like insulin, but it's not at all like it chemically. But it can do something that insulin itself can do. So it can promote glucose transport into tissues. That's very important in functioning of some tissues. For adipocytes, fat tissue, skeletal muscle. It's important in the liver, but it doesn't affect the glucose transport. And it doesn't even seem to do that also in the brain, yet it's very important. It influences biosynthesis of fats and glycogen and protein and DNA, and it has many, many functions. And in the brain, besides doing metabolism, it seems to be important for learning and memory. So many of these processes that we're going to talk about that are influenced by insulin are caused by what we call protein phosphorylation. I was very lucky when I was a student. I started my PhD at the University of Washington, and I was working with two professors who were really at the beginning of their work on protein phosphorylation. And later, they received the Nobel Prize. And one of those people today, I just visited, now has Alzheimer's disease. So you can touch anybody. But I was so lucky to get involved with that. And of course, I met my wife there, and it was a wonderful time in Seattle.
By the way, Don just celebrated his 50th wedding anniversary with his wife.
Hey. Thank you.
Graduate school is lots of fun.
You're right. So what protein phosphorylation is, it's a biochemical process by which phosphate is incorporated into the protein. It's really important in metabolism, but again, a small amount does one thing, but too much is another problem. And we know this is a case in Alzheimer's disease. Excessive phosphorylation of a protein in the brain leads to what we call tangles and eventually the death of the neuron. There's other factors associated with that, but too much phosphorylation in the case of the Alzheimer's disease is something bad. Now, can cinnamon, like insulin, change phosphorylation reaction? So that's what we introduced first in the work we did at Iowa State. So we're going to talk a little bit about that. Okay, time for a movie. Now, if I turn the lights out, and I never allowed that to happen when I was lecturing because I knew what would happen. So we'll keep the lights on. And this title is from my wife, A Reason to Season. Oh, okay? Oh, you're going to do the whole thing? Okay. It looks like you need it. Okay.
I'll take your direction.
Take my next slide. So here are the stars in this. We have cinnamon up at the top. We got insulin. We got glucose. We got a fat cell. And we got what's called an insulin receptor. And here's a insulin receptor. It's got a P associated with it. That's the phosphate bound to it. And then here's a little truck. You like trucks, right? That's a glucose transporter. Okay, so let's take a look at what might be happening here. Okay, here's our fat cell. Here's the insulin receptor. Here's this truck. And here's the glucose. Okay, let's go on now. The glucose would like to get into this cell. Oh, bang. It will come next. Just keep doing it, yeah. And just go ahead, just go on. Here comes insulin now. It's going to bind to the receptor. Hey, now it's phosphorylated. Series of things are going to happen in this cell. Lots of things are happening to allow this transporter now to come to the membrane. And what's going to happen next? There comes the glucose into the cell. Truck. Oh, not yet. That's all right. That's fine. That's fine. Keep going. You're doing fine. Truck goes into place. Now, here comes cinnamon. Hey, now the first thing you're going to see is that cinnamon went to a different place. And this is really important. We found this out, that it can do, you'll see what insulin does, but it doesn't do it exactly the same way. So there it's binding to, oh, there it goes, the phosphorylation. And now what's going to happen next? Here goes the truck. Here comes glucose. And in here, off comes the cinnamon, phosphate. The truck goes back, and that's the end of it. But now, does this really happen? So we'll show you a picture. So I'm going to just show you one research result. And actually, all you're going to need to know is that in this slide, if you have a dark, that means that phosphorus, that phosphate has been attached to the protein. And the control is blank. There's no phosphate on this protein. You put an insulin, black. Put a different amount of insulin, also black, a little bit more. But lo and behold, here comes cinnamon, alone, no insulin present, doing the same thing that insulin itself did. It starts to cascade what we call the insulin signaling pathway in the cell. And now we'll turn to one more thing about glycogen, where someone talked about running. Lots of people who do long running like to prepare themselves for this. Particularly if you do a marathon, people talk about hitting the wall. That means that it comes at a certain time, you run out of your glycogen. That's it. And now you're going to really use some other energy source. You're going to notice the difference. Of course, I knew the difference long before I ever hit the 20-mile mark myself. I had problems very early in the run. So that didn't really happen to me. But glycogen is a storage form of carbohydrate, of glucose in cells. We keep it that way until we need it for our energy purposes. Then it's degraded and gives us energy through metabolism, make a lot of ATP that we need for muscle contraction. Now, insulin or cinnamon alone can increase glycogen content. We didn't show you that, but that was one of the results of this initial phosphorylation and then a cascade of events. But the interesting thing we found was together, they were much more effective than the sum of their individual effects. So that would mean that we get a synergism. A little bit of one thing and a little bit of the other together is much better than the sum of their individual effects. Anyway, this is a result not of ours, but again of Richard Anderson and a group in Pakistan, where they looked at, with type 2 diabetics, what would be the effect of taking different amounts of cinnamon from China, Cinnamonium cassia. Okay, so they put this into capsules, and some people got 1 gram of that a day, some got 3 grams a day, some got 6 grams a day. And you would take that somewhere during the day. And this is the result. So here on the bottom is the amounts of the cinnamon were taken. 60 people were in this trial, 30 males and 30 female type 2 diabetics for 40 days. The yellow of all these different groups shows what their fasting blood glucose was to begin with. Very high, well over 200. After the 40 days, those that got 1 gram, you say, wow, way down to 157. 3 grams, 170, 166. 1 gram is as good as 6 grams. It's incredible. Now, the important thing is what happens if you didn't give them the cinnamon, gave them a placebo. And you see, they were still high. They should have been high because they're type 1s, but nothing happened. Absolutely nothing happened. And the same goes through if we look at triglycerides. Again, the yellow, very high, and then you see going down. Placebo, no effect. Cholesterol. Again, significant differences were seen after taking. But 1 gram is just as good. So why would you want to take a lot if one does this? Maybe you don't even need maybe this much. Nobody has done this experiment. I always like to take the smallest amount that works, because who knows? I mean, yes, people use cinnamon in their food and all that, and it's fine, all that, but no, not everybody takes 6 grams a day for the rest of their life, But again, the placebo. Okay, so we didn't see the change. So you don't have to do any statistics here at all, though I'm sure they did. Just seeing the result, you can see how clear. This was a fantastic result. So now I want to talk a little about the connection between diabetes and Alzheimer's disease. Dr. Alzheimer's first patient is shown here. Oh, went too far. Okay. And there's a picture of Ronald Reagan. So now I'm going to tell you about actually some work that I've been doing in UCSB with Dr. Liu and his students, and now some of mine, on this subject of using cinnamon in Alzheimer's disease. This is on the bottom of the slide is a statement that came from the Alzheimer's Association. It said every 72 seconds, someone in America develops Alzheimer's disease. Last week, there was somebody that actually came from the Alzheimer's Association, talked about prevalence of Alzheimer's in the whole world. So she said it's every 7 seconds. It's incredible. And what you see in this slide is actually the effects of age. It seems like when you get older, you're more at risk of getting Alzheimer's disease, and it's like almost 1 in 2 over 85. That doesn't mean you're not safe over here, but their age is certainly a risk factor. It's not the only risk factor. Yes?
Why, what has been brought into this chart, the fact that the amount of people who are over 85 are much less in quantity than those who are 75 to 84? In other words, what I'm saying, per capita, how many are all over 85 as opposed to those?
Oh, sure, that's right. There's much less. But those that are over 85, whatever that number is, half of those have a good chance of getting the disease. That's how that's calculated. So here it just actually shows, how people have been aged or are over 65 since 1900. And we see now, a big change that occurring. This is from America. That's expected here. And that has to do with the so-called baby boomers, the births of children after World War II from 1947 up to 1964. And somehow that's what they call the baby boomer age. And now it seems like the baby boomers, as it was just reported in the internet a couple of days back, are the ones that are really going to be at risk. And so we'll go right down to the bottom, since we're talking about baby boomers. 10 million American baby boomers face Alzheimer's disease. That's coming up right now. 1 out of 8 boomers. Presently, there's 5.2 million people in America that have Alzheimer's disease. But there are a number of people under age 65 have this problem. Unfortunately, there doesn't seem to be enough money to take care of all these people. And you can see what a problem is already with Medicare. Cost for people with Alzheimer's disease and other dementias is 3 times that for Medicare recipients. Medicare spent $91 billion in 2005. $189 billion is expected by 2015. And it's just going to go on and on and on. And unfortunately, federal budget for research is not keeping up with this, and research money for Alzheimer's disease has actually gone down. So that's pretty sad commentary. We need money to do research, to get answers, to help people with this terrible disease. And as people, we're going to face this situation in society. We're going to live longer lives. We're going to have diseases that we didn't have to face so often. And Alzheimer's, diabetes, are heart diseases just like that. So now, this is what I want to talk a bit about, is what happens in the brain in the Alzheimer's patient. And the only real information we really have is actually after the autopsy, after death of the Alzheimer's patient. Right now, there's no real good diagnostic to show what's really happening. There are some blood tests people are working on, still not really well established how you can tell. So all the tests that are done to tell if someone has Alzheimer's disease are psychological tests and other things like that that relate to memory, etc. But the disease, in autopsy, there's two hallmarks of the disease. One's called the senile plaques, and they're made of a protein or peptide called beta amyloid. And I'll show you a picture in a minute. And the other one is the neurofibril tangles made up of the protein tau. This is something that happens inside the cell. This is something that happens outside the cell. Now I'll show you sort of a microscopic analysis of a brain slice. There we go. Okay. So on the far right, we see a normal neuron. And then when we look up above, it's neuron with tangles. Okay, very good. Okay. Okay. So this is a stain, and this is actually staining that protein tau. It's going throughout the whole neuron. You don't see that in this one because it's only when it's tangled do we get this response. So that's a terrible thing that's happening, and it's going to affect how neurons connect with other neurons. And if they don't connect properly, then you get no neurotransmission. And more than that, the neuron's going to die when this happens. But even after it dies and the cell disappears, you don't get rid of this material. That's still stuck in your brain. And that can also cause a problem, it seems. You're not getting rid of the tangled stuff. That's one of the problems. It's like garbage you can't get rid of. And here's these plaques, which are these. This is extracellular. You can almost see in between. You can see some of these other ones that came from the inside. Those are these here. But these other guys are these beta amyloids. And these things are all entangled. It's all these unstructured proteins making wrong kinds of structures. And this is characteristic not just of Alzheimer's disease, it's characteristic of Parkinson's disease. It's characteristic of the prions in the mad cow disease, in the hunting disease. All of these things have in common making these wrong structures, these wrong aggregates, these fibers, which are happening and associated with the abnormality. How exactly? That's the question. But that's kind of considered not a terribly good thing. So now a little bit about diabetes and Alzheimer's disease. Now this is interesting because many years ago, and well, it's not that many years ago, because the insulin didn't affect transport of glucose in the brain, people thought that insulin wasn't important in the brain at all. But then they found, just like I showed you in our movie, there was an insulin receptor. They found that the brain had insulin receptors all over. And wow, well, if they got a receptor, that must mean there's probably something going on here with insulin. And that's now the case that they find is that those receptors are there for insulin. And in the insulin brain, when you have deficiency of insulin in the brain, this will lead to the death of the neurons. Insulin is found to decrease in the Alzheimer's brain. Therefore, the metabolism isn't working correctly, and all the things that are involved from that, including the aspects of memory and learning, are going to be compromised. But I'll say, okay, then let's just give them more insulin. Well, you know what? That's not a terribly good thing. More insulin doesn't seem to work. It doesn't seem to work like it does with a type 1 diabetics. We've got almost kind of, again, like we're facing like an insulin resistance of some sort. And the other thing is, that's very interesting, is these beta amyloids, they can be cleared in some way by an enzyme that acts on insulin. But if you give too much insulin, it blocks the process that's there naturally to get rid of those beta amyloid plaques. So insulin may be acting in one way, but if you give too much, it does something else and say, hey, if I don't get rid of those plaques, I've got the problem because that's going to affect the tau, and I'm going to have the neuron die. So other people are now saying, and scientists are saying, we don't need insulin we need a modulator of insulin. Well, I'm getting pretty excited now because we're thinking, about sensitizing insulin and what cinnamon compounds might be able to do. Okay, so the insulin in the brain. Okay, so this is some very interesting work from a laboratory at Brown University. Tangles in the brain and their death appear to be related to abnormalities in insulin signaling. We didn't talk about that because there was too many things that are happening. I just showed you the truck moving, but there was a lot of proteins being changed, lots of things inside the cell being changed for that to work properly. And I said, now I talked about insulin may not be useful therapy because of the side effects. Oh, and this is what she raised, and this is an interesting question. It's not really accepted by everyone, but the idea of a dysfunction of insulin is accepted. She calls this actually Alzheimer's disease, diabetes type 3. Well, we don't know. Maybe that's not the case, but there actually are subclasses of all these things. There's not one like type 2 is not always one simple thing either. So it's okay, but anyway, it's something like, the insulin isn't working right. So then might cinnamon, a modulator of insulin action, help overcome defects in the disease state? So we'll just now go and look at a few results on this. Oh, yeah, before that. All right, so we start out with a stick, maybe like that or some other sticks like that, or we get a powder. And we know that the material is in there, but we know it's water soluble. And we want to make this the best way you can possibly make it. We want to make it with the mildest conditions. We don't want to destroy what's there. So we've worked very hard at trying to make a good material that has insulin-like activity. So we start out with this brown material, and we end up with this material here in a tube from this water extract. And it's a nice pale yellow tan color. And we're really delighted to have this because I was analyzing a lot of material that was being sold and it was just brown and icky stuff, This was the stuff this is water soluble, and it came from a water solution, and we can take all of this and nicely dissolve it. But a lot of stuff that actually, surprising, that's on the market that says it's a water extract won't dissolve again in water. And it doesn't even have some of the compounds that were initially there. But yet you're buying some of that stuff. We're just doing research and we're not selling anything, but we're somewhat pleased to have a product like this. So now we're going to talk about an activity that these cinnamon can have. And here we're actually going to talk about not the purified compound, which I showed you on the slide, but just the extract of cinnamon itself. Something that could come about by just taking a cinnamon stick, heating it up in reasonably good water, not the worst junky water, but that actually will release the bioactive materials. So we were looking at this process of something that might interrupt the formation of these filaments, these aggregates, which we believe are, leading to toxicity. So can cinnamon block that? Well, the experiments I'm going to show you now are kind of in a test tube situation. We'll go a little bit further than that after this, but they'll just show you the result here is that when we had no cinnamon in here, we got a lot of filaments being formed over time, or a lot of aggregation. We could see this in our spectrophotometer, that something was happening. There was some turbidity coming, and we didn't really at this point define what it was, but we could see that the cinnamon was interrupting that. How we learned really what was happening is we did electron microscopy on the samples at the beginning and at the end. And this just shows you kind of the result of the electron microscopy of with and without the cinnamon. So without the cinnamon, where we had this increase in light scattering seen in the spectrophotometer, we see now we get these kind of filamentous structures. These are structures that were kind of defined earlier in other studies, but not, so we knew that we were going to make some filaments, and we were pleased that we did, but what we're most excited is that even though we had some other kind of structures here, there were nothing that were filamentous. They may have had some scattering of light because there are some particles here, but it was anything like that. So it was blocking the filaments. And then the next slide shows what might happen, and this has been done in a couple of different ways, but here we start out with at 0 time, and at 24 hours we got some filaments. At this time, this student Dylan Peterson in Dr. Liu's laboratory added cinnamon to it, and let the other one just go on with water. And you see these now, which were formed, disappeared again to this material. And it went on to make more filaments. We know actually now pretty much that not only will prevent the formation of the filaments, but it actually will take apart the filaments. Now what I'm not telling you about, and it's a very important thing, is because this protein has a natural function. Its natural function is to interact with another protein called tubulin. So this protein, this unraveled protein, that's tau is, which is much like the protein in your saliva. It's unraveled, it has this proline in it, will bind with these compounds and affect how it associates with itself. But it doesn't affect how it associates with its natural partner, tubulin. And that's important because that's the normal function of the tau. It has to promote the tubulin association, and that's really important how neurons connect, is this polymerization of this protein called tubulin. So it preserves it doesn't interfere, that is, with what normally happens by the protein, but it does overcome something that's not natural. So that's kind of where we are in results that I'm going to show tonight. We can have lots of questions if you like. I'll stay as long as you'd like. Okay. But then we have one more slide to show. 2 more slides, actually.
There you go.
Oh, before that. Okay, so new directions at UCSB. Well, first we showed you that cinnamon extract, purified compounds, seem to block a process associated with Alzheimer's disease. That's in a test tube. We're talking a long stretch. So now we've turned to neurons. Neurons from the hippocampus of rats. The hippocampus is the seat of where these plaques begin, these tau aggregation. It's the part of the brain that's associated with memory. This is where the Alzheimer tangles begin. But through times, you'll see in this brochure, you get tangles throughout your whole brain. So we wanted to look at neurons now to see whether cinnamon compounds can interrupt processes associated with pathology. We're not exactly looking at the same thing, and I can't really give you the results because it's all preliminary at this point, but we're very encouraged because it looks like it can overcome effects that are associated with pathology of the tau protein. So that's where we are now, but then what's the next step? Well, we probably need to do experiments with animals because we need to know, can we get something? We're trying to find the smallest molecule, smallest structure that has this bioactivity in cinnamon, because there is this issue about substances have to pass through into the brain. It has to pass through the blood-brain barrier. We know some of these molecules that can pass the blood-brain barrier, and we're testing. They're found in cinnamon, and we're finding that, let's say, some changes can take place with these, but it looks like they are bioactive as well. So we're kind of excited, but we don't know yet whether we try these with animals, whether it'll work. But if it does work, then we're ready to say, maybe we can come up with something that would be useful to mankind. And it takes a long time to get there, and we're a lot of us working with very little money, but we're doing the best we can, and we're delighted to have had support from Cottage Hospital. They've given me a couple of grants. I've now granted into the Alzheimer's Association. I hope that they'll look good on that, but today it's very difficult to get money. There's good scientists all over, that aren't able to get funding. And I have 2 of my own sons in the same business, and they're night and day working, writing grants and trying to survive. And it comes down to something like only 10% of the people, that apply can get money. That doesn't mean that 90% of those others aren't any good. It's just so difficult to get it. So we hope that things will change in America, to give more support to good science so we can keep up and tackle these important problems. So thank you very much for your attention. Sorry for the confusion going back and forth.
If, I'm sure that, two things. Dr. Graves, I'm sure, would like to answer any questions that any of you might have on things. I did want to let you know also, and I really appreciate the information that he's brought to us today, because much of it is obviously beneficial. I mean, cinnamon, as Dr. Graves has indicated, is an opportunity for us to help not only prevent type 2 diabetes, but also maybe a path to the prevention of Alzheimer's disease, which is facing many of us who are maybe not quite at that stage, but are laying the foundation today for what can happen tomorrow. So really important with that. I did want to mention also that, and I wanted to use this lecture today to kick off a program that we're interested in doing, which is to look at diabetes prevention and education in the Life Fitness Center. So after probably the spring break, we'll be introducing some things. We have a deal that we've worked out with Quest Laboratories here in Santa Barbara to do very low-cost blood draws for folks if they are interested in finding out what your fasting glucose levels are, fasting triglycerides, cholesterol, etc., and then looking at metabolic syndrome, which is a precursor, again, to type 2 diabetes. So be on the lookout in the fitness center for that information, but we'd like to be able to assist any of you in determining whether you might be on that pathway, and if so, how you can look to prevent these illnesses before they begin. So any of you that have questions for Dr. Graves?
Can I just make one comment? Yeah. Just because there is research in regard to metabolic syndrome, and some of that's been published by Richard Anderson, is actually you yourself becoming, by doing some things to your body that maybe shouldn't happen, or experiencing that, things that can affect you long term. You know, you don't want, you've got to take care of yourself now. Everybody takes care of it to keep from getting to these extremes, like no high blood glucose, no high blood pressure, you know, as much as you can, because eventually it's going to catch up. Yeah. So, but not that you might be able to recover that, but I mean, you don't want that to, you know, to happen if you can avoid it.
Absolutely not. For those of you that had not signed in and that sort of thing, we'll do that after we do questions and answers.
This fellow in the back here at first had a question. Yeah, you now go on.
How do you take cinnamon and how do you get a gram? Like I like graham crackers, you know, with cinnamon. I mean,
well, we take it.
A whole bunch of those. Is that good? You know?
Well, I tell you what we do, and what was done in the trial was in a capsule, and you know we make our own capsules, you know, and there's easy kits to do that. The point is that yeah, you can do it, you know, like you do it, and that probably will help. You can make a drink. And that's probably maybe even a little bit better. But if you take it in a capsule, it bypasses the saliva. And when and the protein's in the saliva, and that sequesters the tannin. If you and we find a lot of times with animals, we didn't get the response we hoped for. Where others, when they force-fed animals, put that right into their stomach, they got better results. So that the binding of this cinnamon on the protein compromises its effects. So you may want to put on your crackers. It tastes nice. It smells nice. But it might be better to take it in a capsule. Yes?
Aren't there different kinds, I mean, different value on cinnamon you can buy?
Yeah. Well, you mean there are different types of cinnamon?
Yeah.
Yeah, there's different types of cinnamon. And what do you mean, the different effects of them?
No, I mean, should you be looking for a certain type, like the Indonesian or the Ceylon or something?
Well, so far, the experiments we've done, or Dr. Liu and his associates have done, is that in these tests with these tangles, we didn't see any differences in the different cinnamons. I'm not quite convinced yet about all of this, but we don't have any. There's no real good data to say that one is really absolutely better than another one. However, there are things in cinnamons, you know, for example, the Vietnamese and the Chinese and the Indonesian have a lot more oil in them. And some of that is, you know, they have some coumarin in it, and that might, again, be involved with blood thinning. So you got to be, you know, so Ceylon cinnamon doesn't have that. But as you know, the cinnamon, this is an interesting thing also. And this is, I don't know where this is going to take us, but in the extract, we've identified some compounds. But actually, the scent of cinnamon, which you smell, is a small molecule. And guess what? It gets in your brain. And it modifies certain receptors in neurons. And so maybe just smelling cinnamon And I'm not going to have you do it. I'm not going to have you do it. He told me to do that, and I collapsed, you know. But I mean, that's kind of encouraging because of something small like that. Maybe that could work. Yeah, well, now, lots of questions. You had a question first.
I was going to ask you whether you worked with the Ceylon as well as the...
Oh, yeah. We work with all of them. But that stuff that you saw was Ceylon cinnamon, but we work with all of them. Yeah. Okay, yes.
I just wanted to say there's a big bottle of cinnamon capsules at Costco.
Yeah, I know.
If anyone wants it.
That's right. That's true. That's good. Yeah.
They used to have, but they don't anymore.
Yeah, that's right. He said they don't have it anymore.
They canceled it.
Yeah.
Every time Costco gets something good, they cancel it.
Yeah. Yes.
I can't make a capsule. I don't know how to measure it. But you may have answered it by saying we get cinnamon sticks and smell them.
No, I don't know that. I'm not recommending that because we haven't done any experiments like that. You know, we've only done it with soluble material. I can't tell you that. So don't take that. That's going to be your answer.
What can I do? I mean, I'm getting closer to that.
You could just take some cinnamon sticks and heat it up in some nice water and drink it.
Okay, thank you. Good deal.
Good. Or put it in tea, or whatever. Yeah.
You say that Ceylon cinnamon's more expensive.
Yeah, it's a little more expensive. But the Mexican cinnamon, though, that wasn't that expensive, but it's not apparently such a good grade. But I don't know. It looked to me.
The bottle that you buy, that it's cinnamon from Ceylon or somewhere else?
Well, no, in America they're not going to tell you that. Not too often, anyway. I mean, it'll be cinnamon. You don't know what it is. Yeah. And in Europe, you know what it is, because they won't allow that to be told anyway. Yeah.
Where's it readily available in Santa Barbara?
Cinnamon? Yeah. Oh, every place is. You can find them in. I got that from Scolari's. You find them in most of the supermarkets, and in the health stores they've got a lot of things from cinnamon, a lot of cinnamon products, a lot of them. And a lot of them aren't terribly good, but spend a lot of money. Yeah.
You heard that cinnamon can stimulate digestion, and also anything about cinnamon being an antidepressant.
No, I haven't heard about that. No. I don't know about that. I didn't know about digestion. I would have thought maybe it wouldn't. But did you know about that?
That it would stimulate digestion?
Well, cinnamon, I mean, these tannins, if you didn't have the saliva, they interfere with digestion. They do something on your intestinal tract that it irritates it. So it's not terribly. That's why people sometimes take tea to put milk in it to sequester the tannin. So I would have thought it would have interfered somehow with digestion. I don't know about depression, but I do believe I would believe that's true, because cinnamon it's also being thought might be not just with memory and learning, but actually with mental disorders, and schizophrenia has popped up as a possibility effects of cinnamon, or I'm not sure of cinnamon, but of insulin action again. A lot of things going on in your brain with insulin that never were appreciated. This is what's so interesting about doing the science and learning, because it changes so much. 1 year, tell you something, and next year we found something completely different. And
Yeah. Now, what are the Chinese doing today with cinnamon?
Well, they're selling it. There's a woman in Richard Anderson's laboratory had a patent for making the extract, and she sells it to some places. There's a company in Florida. There's a company here. They're making money.
How do you know if you're getting pure and not a lot of filler?
Yeah, you weren't getting it pure. You're getting a lot of stuff that's, well, I mean, if you get pure, if it's whole cinnamon, it's whole cinnamon. If it's a cinnamon extract, you don't know what you're getting.
But the powder is generally
I'm going to tell you one thing. Oh, man, there are lots of questions. I'll tell you one. All right, go ahead. Yeah.
Don, you're giving us a lot of ideas as to what we can start using now. Therefore, in brief, what's the ongoing research going to prove then, or where are you going? I know you said it, but I didn't quite get it. Briefly, what's the new research going on?
Well, there's the research that was done before was with diabetes. Okay, there's nothing that could show that if you take cinnamon in a capsule that'll help you as a diabetic, because we don't know if it'll get through your brain, into your brain. We don't know how well it's going to be absorbed in the gut.
So if we're taking it now,
You're taking it for diabetes.
diabetes, right?
You're taking it for diabetes. Okay, and I guess on the basis of what's been published, you'd say it's probably good.
Now your
But-
research is in Alzheimer's. Then.
Yeah, because I think that we were following up this the lead of insulin action and malfunction, just like it is in the case, to see whether this something that can work on insulin in peripheral tissues may be also do the same thing or something like it in the brain. But we got to find something that we're sure they can get into the brain. How to get it there, and what is it? Yeah.
The tables that you showed in terms of dosage and so forth, and the results of dosage, what cinnamon was used,
Cinnamon. Oh, that's an interesting story.
and how was it taken?
It was taken after meals. The capsule was taken. Capsules were taken, right? And it was cinnamonium cassia from China. But that took a long time to learn that. You know, for a while they wouldn't get the pills. And when they gave them to us, we analyzed it by our mass spectrometry techniques, and we could tell. We could tell the different types of cinnamon. There's certain signatures that we know, we look for. And there's no question that was from China. Yeah. Wait, with this woman, I had a question. Haven't had a chance.
Can you eat too much cinnamon? Are there any negative effects of
I would think so. I would be afraid to take too much of anything. I don't know. I don't like that idea, especially, with the ones from China where they got a lot of coumarin in that. People, you can't, taking some other medications. I'd be a little bit careful. Take as little. I don't take that much myself. I'm afraid of it. My wife takes more than I take. I should take more. I don't take care of myself. Well, I take about a capsule a day. That's all I take. It's not even a gram. Half a gram, maybe.
In the ground up form, is Ceylon cinnamon, is there any color difference between that and other cinnamons?
Yeah, I mean, it's going to be lighter. It's lighter. But not that much difference. But it's also, it's not. These things aren't completely stable, and some will darken earlier than others in the powder form. You got to be careful with that. Yeah.
You do want to caution people, though, if they are diabetic and taking insulin,
Yeah.
that you add cinnamon to the mix, you can become hypoglycemic.
Yeah.
You really have to.
Good for you. You know what? I never tell anybody how much to take. Because I'm afraid of that. They ask me, and I said, well, Richard Anderson recommends this, but I don't want to do it. I'm not a doctor, and I don't know what you're taking. But you can't imagine how many telephone calls you get. That's all they want to know, basically, is how much should I take and which one should I take? And I don't have answers for either one. But, I mean, we can kind of say something, but, we can't really. That's a very good point. I have to be really careful. I get scared from this. I don't want to have somebody that's taking something and something else happens. Say, you told me to do this. Well, thank you very much. Okay, one more question then.
So, was there a comment about the overdose?
Yeah, taking too much.
In the cinnamon, would that indicate that, like a person who already has Alzheimer's couldn't benefit from taking cinnamon?
Well, I don't know. I mean, we haven't done anything with anybody with Alzheimer's at this point. I just in general, you could see the result with diabetes. Why would you want to use any more than 6 if one did it? Why wouldn't you even use less and find out? Thank you very much.